What I'm going to do today, I hope, is to basically, this is a research seminar to tell you about the latest work that mostly done by a postdoc who has just gone back to Japan, and we'll come to that. But I want to introduce it, and it's sort of partly about results, it's partly about ideas, and it's also, I hope, can show you that actually a rather simple experiment can shed fresh light on old, almost not appreciated problems. So the talk is called Switching Mitosis On and Off. Sometimes I just call it Getting In and Out of Mitosis. And I'll start with just a general introduction to try and put things in perspective. So as you all know, the cell cycle is about cells growing and dividing. Very simple. Expressed in the cover of this book, which is almost the first time we see the phrase cell cycle in print. Before that, people tended to talk about the cell division cycle, and it's not such an old concept, actually. I mean, that is to say, people in the olden days, old biologists, didn't even realize this thing existed, even though they knew about the existence of cells, because they really didn't know what went on inside the cells. And from my perspective, there are really only sort of three interesting places in the cell cycle. Now, you probably have your own favorite places, but mine are particularly these switches here, the switch of getting into mitosis and getting out of mitosis. There are, of course, some other very, very important switches, like the growth of the cell throughout the cell cycle, and what a lot of people focus on, the initiation of DNA replication. But I just want to focus on these two places. Now, I say these are switches. Not absolutely clear that's really true, because exactly where in this process of getting in and getting out of mitosis do the two switches, which I've indicated here, take place? They're more continuous. I mean, at some point the spindle pole body goes from 1 to 2. At some point the chromosomes start to condense. At some point the nuclear envelope breaks down. At some point the chromosomes suddenly come apart. At some point the cell actually divides into two. It's more of a continuum. Yet what my interest is exactly in the switches. What happens when things change? This is a very old obsession of the Greek philosophers. You may remember there was a wonderful, my favorite Greek philosopher was called Parmenides. And Parmenides, we don't know exactly what he thought, really, but he said, what is, and what is not, is not. And it's kind of hard to argue with that. But then he says, well, if that's the case, how can what is comes to be what is not? And how can what is not come to be what is? So actually change is impossible, he argued. And he said that all this things changing all the time is just an artifact of the human imagination, and really the world is a homogeneous, changeless sphere. And I still think that's a sort of very interesting concept. And of course there's a sense in which he's right. I mean, matter is neither, I guess, well, no, let's not go there. Anyway, so it is interesting to know, I mean, and that's the most interesting thing in biology, I think, when things change. It's much harder to study a very important problem for cancer, which is when things stay the same. Because if you think about it, most things in our bodies actually stay amazingly much the same. The shape of your nose doesn't alter much in the course of your life, and it's a good thing it doesn't keep growing. It's also a good thing it doesn't sort of shrink away. It just stays the same. And that's, cancers come when things just grow a little bit faster than they should. But this is not the topic of the lecture, and I don't know anything about it, and I don't understand it, but I think it's very important. And if you're young and looking for a research project, I suggest that you think about trying to study how things stay the same. It's a really good project. So this is a pretty simple switch, and it's just for turning the lights on and off. But behind the switch, it can be quite complicated. And I would remind you that we biologists are a pretty naive and hopeless lot, and our methods for analyzing this would be to remove that spring there by making a knockout switch mutant and asking what goes wrong, right? Very curious. I mean, you know, it's pretty... whereas if you were an engineer, you would say, well, I have to put that spring in there because otherwise it won't feel positive enough. You won't get a nice, satisfying click out of it or something like that. But we're reverse engineers. We work backwards, so it's tough. So here's an example of a switch in actual action. These are a beautiful movie by Peter Lennart of maturing starfish oocytes. And you see, I mean, let me just run this again. Look at this. I mean, it happens in the space, this is speeded up, of course, but this happens in the course of about 5 minutes or something like that. You can almost see it happen with the naked eye. And it only happens when these particular oocytes see a particular hormone. It's 1-methyladenine in this case. And let me show you another marvelous movie made by Peter. This is extraordinary. This is the same thing from inside and now color. There are the chromosomes, and they line up, and then... Fantastic, eh? And that's all switched on by this hormone. So it's the same thing with, but except much less spectacular. You can't make movies of it because the frog eggs are totally opaque, so it's very difficult to see what goes on inside. But progesterone causes this switch. This was a movie, actually, and the formation of that white spot on the dark hemisphere is what's known as oocyte maturation. And it was a long time ago, 1971, shortly after I started to be I was a postdoc at the time. And these guys working in America found that what progesterone did was to cause the activation of an inactive precursor to bring about those changes that I've shown you in the movies. I mean, it's exactly the same. You know, the nucleus breaks down and a polar body comes out. And they called this substance, this stuff, MPF, and they knew MPF existed because if you took a mature oocyte, which you also call an egg, and injected the protein from the egg into a fresh oocyte, the oocyte behaved exactly as though it had seen progesterone. And it was discovered, these people discovered, that number one, the factor was heat labile, it was protease sensitive, and it seemed to have the property of turning itself on, which was also extremely intriguing, a sort of autocatalytic activation mechanism. But they did not succeed in purifying it because it proved to be extremely labile.